Okay, so we have 10 minutes to start entering into 2 or 3 classes of the nucleus organization. And so let's not discuss. So the nucleus organization is a big story. It involves now quite a bit of experimental group from genetics, microscopy, to really characterize how the chromatin inside the nucleus is organized and the normal and pathological condition under perturbations. So modeling chromatin organization. So for this, I would like to start with questions about polymer physics. What seems surprising in this entire field is that the simplest of all the polymers gave the best, is one of the best models for interpretation of data. So part 1, which is really, so the introduction is really about how the nucleus is organized, So you have your cell, inside you have the nucleus, and again you have chromosomes. So in mammalian cells, people have described territories. That is, if you have ever done simulations with a polymer, and the polymer repel each other, that is, or if you cook spaghettis at the beginning, or it depends what kind of spaghettis, but basically you can see that they stay. This is what they see under the microscope and the interpretation, is that you have territories. The chromosomes do not mix for some reason that are unclear. So the question was turned to the physicist: is it true that if you simulate a polymer with exclusion forces, I mean like Lennard-Jones forces, that naturally gets segregated this way? Of course, it's not true in yeast. In yeast, there is no segregation. In yeast, what you have, if you look at the nucleus, so this is the nucleus. Now this is the nucleus of the yeast. I'm not going to draw the cytoplasm. And you don't have this. You have, this is the centromere, as we've drawn before, and you have the telomeres. And the telomere form clusters attached to the boundary. Under some conditions, the telomere can even detach and form, in essence, like a big cluster. So there is a big debate about how the nucleus is organized. And so I have to tell you about one more type of experiment. So there are 2 types of experiments. This is the description of where the data will come from for this interpretation. What can you do to see what happens inside the nucleus? Well, using microscopy, people have tagged chromosome, and they look under the microscope, and what you see, and you can collect trajectories. Now the question is, what can you extract from the trajectories? What can you extract? What can you extract from trajectories? So this is a big debate. And I mean, debate means you can extract the fusion coefficient, but we're going to see we can extract much more. You can extract some forces. You can extract information about the chromatin, about the polymer itself. You can extract information about the alpha of the anomalous exponent, let's say, before you do anything, and after you induce a break. So suppose now you do the same experiment, now you have a break here. Can you see the effect of the break in the trajectories? The answer is yes. Now we have a paper. We are trying to get this paper published. We're doing a mix of experiments. Not that we did experiments, but we analyze the data to try to understand how do you differentiate trajectories, what kind of statistical information you get before and after a break.
Sorry, break, you mean like you stop the measurement?
Break means you do a break. You cut double-strand DNA break. So you have your DNA. and then you cut it like this. It's really a break.
Okay, sorry.
Single-strand DNA break is much easier to repair, but double-stranded DNA break, this is much harder. There are different two pathways. I don't want to describe them now. Basically, that you glue them, or if you have missed some information, you have to find a template to get back the information. And this is much harder because then you are involved, the DNA or the chromatin is involved in the search process. So again, you don't search with a Brownian particle like we did in chemical reaction to search for a target. You search like the head of a snake is searching, and it drags its entire body toward the target. Okay, so this is one type of data, which are called, so the first type of data I've described, this is called single particle trajectories data, which give dynamical information. So people perturbed, you can do genetic information, knockout, you perturb some genes, you perturbed, and then you go back to the trajectories, try to extract features, and then you compare features before and after. So this is the first type of data. This is called single particle trajectories. This is what we just described now. The second type of data that is going to be very interesting, and that has been the result of quite a lot of effort, is to have a frozen picture. So you have your DNA, you freeze it, and then you look at the following question. Can you obtain, fix, let's say, monomer, let's call it I, and fix another guy, J. Average, so get information about the distance between I and J, the encounter probability, and do that over all I and all J. Basically, you end up with a distance or a frequency matrix of, so you get your, let's say, let me write it as a distance here because this is clear. Now you get a matrix, and if you discretize this with a certain scale that you fix, you can end up, you can get in the end a matrix of 1 million by 1 million. And you do that, you average over this over many cell realization. So what you end up with, what is called the frequency of encounter. That is, you look at the fraction in the ensemble of cells that I was close to J in, let's say, 1 million cells, and you look at this frequency, and you do that for I and J. Now, the big question in the field, what can you reconstruct from this matrix? So what can be reconstructed? What can be reconstructed from the encounter probability matrix? This is unsolved. Probably there are many solutions. Probably it's completely nonsense because you average over many cell realization, and if the cells are in different configurations, you are going to have some kind of ensemble. What can you get from this ensemble? Well, it turned out that people have discovered many things with this. They discovered that they are part of the DNA, or part of the chromatin, that they are forming subregion, and this is stable. So then you can try to reconstruct this subregion, why this subregion is stable, why it is together, and get more information about how the nucleus is organized. Again, for example, in yeast, if a gene is expressed, it can be on the surface. If it's repressed, it can be inside. So there is also a relationship between geometrical organization and gene expression. So now, the first approach let's spend 5 minutes on this, and so this is really part 2, which is what kind of polymer model are we going to use to describe, to analyze, to calculate. So for example, the type of question you would like to be interested in is, for example, as a function of distance here. If you let everything evolve according to Brownian motion, how long do you have to wait to form a loop? Right? So let's ask some questions. So the questions that were of interest that I would like to discuss, and for this, we need a polymer, we need a model for that. So the question is, what is the mean time to form a loop? Form a loop means by Brownian motion. Second, we are going to discuss is when you have the mean square displacement, which is the average, let's say, of xt plus delta t minus xt, and you average squared over your realization, it goes to delta t to some power alpha. What is the meaning of alpha?
David, can I ask, what do you mean by a loop? Do you mean with your diagram where I and j come together?
Yeah. So a loop. So suppose you have a polymer. Suppose let's just discuss about the two end A and B. How long it would take for B to come in the ball of, let's say, radius epsilon, and epsilon you can vary, but this is something that it's supposed it is fixed. How long it takes for this guy to enter into this loop for the first time? Right? And can you write an expression? Can you find it analytically?
Okay, is that how you construct it, or are you thinking of two epsilon regions around A and B and then coming together?
That's more or less the same. If you can solve this one, you are happy. And it, of course, I put the end, but it can also be inside, as you can have in here. How long it takes for this guy to come in this close proximity for the first time, and then it separates again. So to have this will give us the rate of the information about the encounter probability, the encounter frequency matrix. But now we want to have this using a model, using a polymer model. So we know how to solve this for Rouse polymer. What is beautiful is that nature seems to be using, I mean, the Rouse polymer seems to fit quite well, although it has some little discrepancy, but it's very good in terms of analyzing the chromatin at the scale that we have under the microscope. So what is the meaning of alpha? And then there is also how do you construct basically estimators for parameters you want to extract. For example, how to extract from information, how to extract the diffusion coefficient, of course alpha. You can extract also, and this we will see, forces, and I'll explain how to do this. That will characterize the motion of the chromatin or the motion of a single locus. And so for this, to answer this question, so this will take us at least, I think, 2 classes. We need to introduce a model, which is the Rouse model. It's
So could I just ask so I mean, we know that the DNA can wrap around histone complexes, and then these things can separate, and this is one of the mechanisms in epigenetics, right? So have you thought about these things?
Well, nucleosomes, this is the scale of this is really the scale where nucleosomes are not distinguished.
Yeah, no, I can see.
Because then if you want to add nucleosome on a polymer model, this is very interesting. This is very nice, actually. You have first to have, so let's introduce the polymer model. So here we'll discuss mostly Rouse, although I'll explain other type of polymer. So you have beads that are connected by spring. So you start with bead number 1 and 2 and 3. So this is really, of course, graining of what? What are we talking about here? As you said, you know, David, you're talking nonsense. You have nucleosomes where the polymer, the DNA wrapped around, and then there's another one, and then the other one, and then it goes like this. And this is what you can see in textbook. It goes twice, and then twice, and then twice. Here, what we're talking about is something abstract. This can represent any number of base pairs. So we have to define what is the scale at which we are going to use this. For example, we can say, I'm going to collapse 3 kilo base pair in each bead. So then I don't see this picture.
Yeah.
If you want to see this picture, this is very hard. Then if you want to make any simulations, people are doing this. It's very hard work to look at simulation. Then it's basically a molecular simulation. You try to look at the interaction, but then you don't go very far with the time scale of your simulation. If you want to have long-range processes such as looping, looping is a rare event. You know, it's the time it takes for two to meet again. So this is a rare event. No chance that if you add, or if you start with this complexity, you answer this question. This is for example, we looked, this is also in published work, when you send UV light, you create damages. The damages turn out to lead to translocation of these nucleosomes. That is, they are pushed away, or maybe they're falling. Now you need a different model. You need to add on this a model for the nucleosomes. Okay, if I have time, I'll discuss a bit this. But so now the idea is how you write now equations for this Rouse polymer. So let's start with this. So this is very classical. In the Smolokhovsky limit of the Langevin equation, so x dot equals square root of 2d. This is a classical stochastic equation for one single particle that is moving with diffusion coefficient d. Now the Rouse polymer is very easy to understand. You have bead xk. It is connected to bead xk minus 1, bead xk plus 1 with a spring constant. And then you write down your stochastic equation for this guy, which is x in the Smolokhovsky limit of the medium. This is, let me write it, 2rk minus rk minus 1 minus rk plus 1 plus square root of 2d. Let's suppose this is the same for all of them. And this is the white noise. And you have to be careful at the end because this guy has no guy on the left and this guy has no guy on the right. Now what do you do with this? Now it's becoming more complicated than the simple stochastic equation for one particle. It turns out that you can do a lot. And so let me explain in a few minutes here how from this you can start now construct polymer with a given anomalous exponent. So I have to tell you that if you look at the anomalous exponent of the monomer, okay, of t squared, and you look, let's suppose that at time 0 this is in 0, this goes like it's t to power 1/2. Diffusion, this is 1, here this is 1/2. So this is the t small, it behaves this way. So now let's see how, I mean, a method of construction of a general polymer. The idea is to start by using a diagonalization of this matrix here. So you can always write this is a matrix. You want to diagonalize this matrix, look at the eigenvalues, and look at this equation in the space, in the eigenspace. So this is a three-diagonal matrix, and this is well known. This has been worked out 40, 50 years ago, that you can use the following transformation. You write another set of coordinates, up, is Rn alpha p of n from n going from 1 to capital N, and alpha pn are given, so this is really an orthogonal matrix. This is 1 over square root n for p equals 0, 2 divided by n, cosine n minus 1/2 pi p divided by n. All right, so this is, you change coordinates. This is the Rn, you multiply by this matrix, and now you want to see what is the stochastic equation satisfied by this UP, so you just replace here. So what do you win by doing this? This is a way to diagonalize the matrix, and you end up with an ensemble of an equation which is d up dt. Just differentiate this with respect to time, equals minus d kappa p up plus square root of 2d, and then you have a new noise, whatever, okay, let's call it wp dt, where kappa p equals 4 kappa sine pi p divided by 2n. This is important, squared. So this is true for p going from 1 to n minus 1. For 0, u0 is really only the center of mass, which I don't care, because basically, if you have a polymer, the polymer will move. So you can take the average of the motion, and this will give you the center of mass. Now, p in the polymer, now you cannot have this translation motion, and this is basically what you have here from 1 to n minus 1. Right before we had something beats from 1 to n, so you can ask where is the missing one. So the missing one is just the Brownian motion of the center of mass, which I'm not writing. So I want to make sure that, yeah. So let me write down. So before I have forgotten, here there was a kappa, which is the spring constant. This is the kappa that is in here.
Don't you get additional vector in the noise term?
Yes.
Because you
yes,
it's
but
an eigenvector of.
That's right, but this is equivalent to another Brownian noise. But you don't care.
Okay.
That's why I wrote it tilde, it's a sum, and it's a Brownian motion, so it stays Brownian motion. What is important is this, and now in the 2 minutes that I have, I just want to explain how we move all of this. So what have we done here is nothing really. This is dUdp. So now I have to write one more thing, which is the so associated to this, there is one important quantity, which is the energy. So the energy of R1, Rn. This is simply the sum of kappa divided by 2, Rn minus 1, squared from 1 to n. And you can always rewrite this using the gradient of u in here, gradient with respect to Rk. Now here is the idea. This is in the Fourier space. I mean, an ensemble of Ornstein-Uhlenbeck equation. This is linear. Kappa p is given. So suppose now instead of 2, we changed it to beta. Beta different than 2. All right, you have the 2 here was crucial. Now let's put another exponent, and now let's do the inverse of this and see what kind of polymer you get. So this is the first part that we will do next week. I want to see what you will get here, and in particular when you do this beta here.
And the reason you're doing that is because that's the fractional eigenvalue of that Laplacian operator. I mean, this is a finite difference.
That's right. That's right.
But it's a Laplacian.
That's right. It's a Laplacian.
And so you take the fractional Laplacian of that, and you get that eigenvalue.
That's right. And now you'll see how this will propagate, and yeah, this will give us the fractional and how it will spread into the energy. And this energy. So if we, I'll take just a minute to write this because as Kevin started to mention, so in when you write this into this U mode, you get 1/2 of the sum of kappa p dp squared. This kappa p is equal to this. Now we're going to change it. So this, let me put it in square. Now we are going to change it to beta. Up can be, you can use now this guy to plug into here. And this is the energy. So when we are going to express everything back into the Rn, this, what is the interpretation of it? This is to explain that the connection is between the two neighbors. But now, when we will expand this, we will get exactly the new matrix, because with the beta, which was 2, you had all this cancellation. Now you don't have any cancellations. Now all of them are going to be coupled. As a matter of fact, you know, you can write it kappa p beta. So up is, let's replace, this is the double sum of r, let's call it nm alpha pn, rm alpha m p. So you have to sum over p over n over m. And now you are not going to get necessarily this, but you're going to get the coefficient in front of Rn and Rm, right? Because this is not going to be anymore, so the coefficients that you're going to get.
So it's not distance dependent anymore?
That's right.
I need to beat.
That's right. Because before, if you look at the coefficient A alpha m, this is sine beta pi p divided by 2n. And then you have to multiply by cosine m minus 1/2 pi p divided by n, multiply by cosine m minus l minus 1/2 pi p n. This is the coefficient that we are going to discuss. Yeah, and you have to sum over p. If you have here squared, everything will cancel. If you don't have, if you have a general beta, it doesn't cancel. So this is a procedure to construct this new interaction. So next week, let's stop here. Next week I'll show this, and then I'll show how you calculate now with this the new anomalous exponent and how this is associated to the beta. So to see the connections.
A quick question. Do you find general decay with the separation ij in that structure? As the separation between I and j increases, the
there
becomes
is
smaller?
That's right. That's the answer to your question.
Exactly.
It decays like very quickly.
Okay.
Yeah, you see. Yeah.
So it has a local sort of...
It's local, yeah, it's local, but you need
okay.
this stuff far
So
away.
it's like a local many-body sort of thing.
Like in many, sorry.
It's like a local many-body effect in a way.
Well, you need what happened far away, otherwise you cannot construct. But it's true that it is small.
Yeah.
Is there,
a local, non-local model.
That's right.
but is there a characteristic length scale too, or is it like in parallel? So
It's okay. It depends what we're talking about. You will see the t to some power alpha. Now, what is interesting here, the correlation function is still exponential.
I see. Cool.
That's the way it is. Okay, thanks.